Hi, let's see if I can get my slides up. There we go. So I want to talk a little bit about the management and analysis of pre-implantation genetic diagnosis data. And my name is Deanne Taylor. I'm an assistant professor in the Department of Obstetrics, Gynecology, and Reproductive Sciences at Robert Wood Johnson Medical School at Rutgers University. I'm also part of the Department of Genetics, and I teach and serve in the Graduate School of Molecular Biosciences. I also wear another hat in that I am the Director of Bioinformatics for the Reproductive Medicine Associates of New Jersey, which includes RMA Genetics, which is a separate company, and I work for both organizations as a bioinformaticist, statistician, and overall data jockey. So I want to talk a little bit about topics in managing data for clinical laboratories. So my topics are going to be data standards and requirements for clinical molecular diagnostic laboratories, what exists now. I also would like to discuss data and pipelines for PGD as a molecular diagnostic. I'd like to talk about next generation sequencing PGD and human genetics and future pre-implantation genetic diagnosis trends. And each of these, the last topic will be a little bit more speculative and a little bit more about human genetics. And the first few topics we'll talk about how data is handled in clinical laboratories. So data management standards in clinical laboratories. Most of us think, at least people who are functioning clinical laboratories, if you're not directly involved with the maintenance and action of a clinical laboratory information system, you think of it as a black box where you have patient metadata, test and instrument data, raw test data from instruments, post-analysis results, and test result reporting all coming out and surrounding the clinical laboratory information management system. And there's some sort of workflow and standard operating procedure that you've established for using that information system. But often people don't understand or are not aware of everything that goes on inside the box, both in how data is managed, how quality is assured, and how the data integrity is maintained. And currently, there aren't actually a lot of regulations around what actually goes inside that black box. So there's a lot of regulations surrounding the information that goes in and comes out. So in CLIA, for those of you who are familiar, these standards were started in 1988, which really outlines regulations and how clinical laboratories are it regulates clinical laboratory activities and reporting. And so some of the data requirements that are explicitly outlined in the CLIA regulations, which I've linked below if you're interested, is things like patient identification, specimen identification, when the data has come into the laboratory, what test was performed, and hopefully you have your tests on record. So what test on the record tests that you do, which test was performed and when the test was done, the results from the data analysis, and information that any specimens that didn't make it into your laboratory because of quality issues. So there's a few others, but that's the main gist of what CLIA requires for clinical reporting. And there's also implicit quality and management requirements in there. For example, there's confidentiality of patient information. Now these implicit quality and data quality and management requirements are implicit that they don't actually tell you what data is required or how to handle that data, but they do require certain actions on the laboratory's part to maintain that information. For example, patient information should be kept confidential, so therefore your database or data store should be secure. Also, you have to securely maintain your test results and your data for about 2 years. It explicitly says 2 years. So you have to have some way of accurately storing it and maintaining that data. You also have to have data on the testing systems that did the analysis. For example, you have to have information in your reagents, your instruments, your materials, and supplies. That's implicit to the regulations. And also you have to have accurate result reporting, and your test reports have to have a certain amount of data in them. And that reporting somehow has to come out of your information management system in a correct and accurate manner to give the correct report to the clinician. But there's no explicit guidance on the quality of the data, and there's no actual regulations that try to do quality control on the data that's within that clinical laboratory information management system. By the way, if you have any questions that you can think of as I go through this, because this is the first time I've given this talk, and this is a talk I've wanted to do for a while, so I'm probably not going to be clear on some things. Feel free to start entering your questions and answers if you're doing this live while I talk, and at the end of the talk I'll get to your questions and answers and interpret them and tell the audience what you're asking and stuff. So one of the accrediting bodies is the College of American Pathologists, and the CAP molecular pathology checklist is used for accreditation. And they have, this is outside, they're kind of a little bit more, they have a specific molecular pathology set of requirements for accreditation on top of CLIA. And recently, they've added standard section on bioinformatics pipelines for next generation sequencing, which is really nice to see. There includes data analysis standards for NGS for next generation sequencing, pipeline documentation, process validation, quality control, data, raw and derived data storage. I'll talk more about this later on. But essentially, especially the version tracking, it's all very nice to see because it's all stuff that wasn't previously required for a lot of laboratory data. However, especially molecular data, pardon me, but however, there's no other guidance really on data management of processes for other bioinformatics heavy platforms, such as microarrays or high complexity PCR experiments, like array PCR experiments. And that doesn't exist. So they've taken this great step for next generation sequencing, but we're still not completely there with the other, with workflows and data management for the other processes that are commonly used in laboratories, especially PGD laboratories. So there's also overall no guidance in CAP on their checklist for quality or best management practices. In New York has a clinical laboratory evaluation process that they produced several documents. It's actually quite rigorous, and we've worked with them because we're in New Jersey and New York State is right next door. So we actually work with the Department of Health there, and they're much more complete in what they require from a laboratory, I found, than some of the other standards that are out there. And I don't know if they're the most detailed of all the states, but they're pretty detailed. So one of the things they do go through laboratory information management system requirements. Some of them they leave it up to the laboratory for exactly what implementation the information management system takes, whether or not it's commercial or in-house. But they do have requirements such as test a reported, archive, and maintain in an accurate and reliable manner. You have to have proper maintenance of your LIMS per the manufacturer. So your LIMS has to be updated according to the standards of the manufacturer. Your peripheral devices like printers and such have to be maintained. Your validation, your LIMS has to be validated prior to use, and you should have it validated in a manner that shows that your tests are accurately reported and data is accurately maintained. Your data has to be backed up and recovered in proper manners. You have to have provisions for the LIMS failure in some sort of backups process, so in case something goes wrong with the LIMS, if the LIMS fails, you should be able to go into the data and access the data outside of the failure of the LIMS. You have to have clinical reporting requirements on amendments. The amendment, not only the original report has to come out, but there's certain requirements for amendments that have to be added to their existing report, so they have that as part of their policy. They also have limited personnel access according to permission, so your LIMS has to have a hierarchical permission system under New York State. And also, you have to have power surge protection, UPS power backup, which is perfectly reasonable to expect. And also, they actually require supervisor review of any manual entries and adequate training for people entering data into the LIMS. So this is pretty good, and we were happy to see a lot of the feedback that we got when we were going through this process. We're coming from people who did some very careful consideration of your system. So they don't rubber stamp things in New York State. They actually go through your process, and they pay careful attention to how things are done. However, even in New York State and across the board, in general, there aren't really explicit requirements for personnel with certified or demonstrated expertise in clinical data management. There's plenty of requirements for what kind of laboratory supervisory experience a laboratory supervisor has to have. and other people with positions of responsibility in the laboratory, in the clinical testing laboratory, but there aren't explicit, that I've been able to see, any kind of requirements for people to have some sort of certification in SQL or laboratory information management or HIPAA, or the people who actually handle this data, including the LIMS manufacturer. There's no certification for the people that do handle the data. There's also no certification process for the information management tools or database systems. There is no certified LIMS system that could be used in a clinical laboratory. That's, again, completely on the shoulders of the lab. And there's no oversight or regulation for good data management practices in the laboratory outside of some of the stipulations in the LIMS system that New York uses. There's no stipulations for demonstrating data integrity month to month, or looking for correctly normalized database tables, or so that the data that you put in last year is still the data you have today. There's no checks on that. And there's no requirements to demonstrate adequate data security, quality, reliable, and reproducibility. And in general, I haven't talked about HIPAA in this talk. I'm going to assume that HIPAA is in place, that you have correct firewalls, and that your data is adequately secure in your environment. But outside of the security and access security and backup kind of security, the kind of security I'm talking about is security for security against mistakes. So I guess it's an integrity security. There's not a lot of regulations or checks in the inspections to make sure that the systems that you put in place to handle this massive amount of data are actually doing what they say they do. So that black box is really the black box. So in quality-driven data management, without external guidance, we are actually we need to establish our own rigorous internal standards and protocols for automated data acquisition. How we do that and how we make sure that it's done accurately. Whether or not we also have to have protocols for automated and periodic tests and reviews for quality assurance in our databases. We do quality control at the point of data entry. So there's tight quality controls, not only in the data that's already in the database, but in what humans put in. So there's supervisory review of our data. There's also quality checks at the point of entry. So people can't put in a crazy date or put in an unexpected value. So those kinds of things have to be, so the quality of the data going into the database is monitored and controlled. Also good data management practices. I go more into that in a little bit, but some example considerations are that your data security, you should have your privacy, proper security, and backup should be in place. Your data integrity and design, you should have properly typed variables in your database. You should have periodic checks for variable ranges and outliers in your data. You should have your database should be properly normalized in the case you're using a relational database, so that you don't have modifications that are not expected. Also, quality control, you should have easily accessible quality control metrics for your data set, and you should also test the performance of your LIMS or your data sets of your workflows to actually see that they accurately produce month to month the same results that you expect them to produce. Also, what software implementation you're using, and whether or not the systems are using have demonstrated and been reviewed and validated on a periodic database. So not just the data quality control, but the systems that generate the data. So these are example considerations for quality-driven data management in production. So most labs have to go through this on their own without guidance or inspection. Again, the black box is in place. So for PGD in specifics, we have three different methods we use for PGD. PGD is essentially where pre-implantation genetic diagnosis is a way of in the laboratory of determining whether or not an embryo is a carrier for a particular disease, or whether or not it has the correct copy number of chromosomes. I'll talk about that in a minute. But for all this kind of measures, we use three main analysis methods right now, at least here at RMA. And one is PCR, which we amplify known sequences and we identify gene disordered carrier statuses or chromosome copy number analysis in embryos. We use microarrays, and a lot of other places do too, especially array CGH, where you can utilize many thousands to about 1 million different individual markers. And these points will give you information on deletions, chromosomal rearrangements, or chromosomal copy number status. And that currently SNP arrays and array CGH are used. And also there's next-gen sequencing, or second-generation sequencing it's often called, and that can be used for identifying gene disorder carrier status or analyzing chromosomal copy number. So the methods in next-gen sequencing could range from targeting specific regions to look at particular carrier or affected status, so looking for specific biomarkers or sequence markers, or also looking for whole genome sequencing and for different types of method like chromosomal copy number. So data size examples for PGD platforms, if you're interested, PCR, you expect 10 megabytes up to 20 megabytes of data maybe if you're storing per sample. That's generous. But in terms of terabytes for 2 years of data storage, it's not very much. But as you move up the range, if you go to microarrays, it's about between 3 to 5 terabytes for 2 years of data. Targeted NGS, when you're looking at small regions of the chromosome. For next generation sequencing, when you're looking for specific markers and you're targeting what sequence you're going to be sequencing, maybe 8 terabytes a year if you're doing 500 embryos a year. But in genomic NGS, if that's where it's going, it's going to be much larger, and you've probably heard about this data sizes in next generation sequencing. And really, depending on how deep and how much sequencing you do per sample, this may be even an underestimate. Having a few gigabases, a few gigabytes of data per embryo is probably not something that would be unusual if you did whole genome sequencing on an embryo. And even with a conservative file size of about 1,000 for about 1 gigabyte of analyzed data size and about 1 gigabyte of compressed data, you're still going to have to deal with about 24 terabytes over 2 years of storage if you were doing all embryos as whole genome sequencing, which may or may not happen. But it's a consideration for data size. So in PGD information, in PGD laboratory settings, we have to assign unique IDs for patient embryos. These are the kinds of things that we're considering to be important for building a laboratory information management system for PGD. We're also require automated tracking of samples through a laboratory. We want detailed records on all reagents and instruments. We need to have data activity and change tracking in the database. We need to have end-to-end process flow control so that we know at all points where the samples are and how the samples are moving through the laboratory, if there's any bottlenecks or when the data is expected to be ready, especially in the cases of fresh embryo transfer. We need clinical reporting to required standards that are laid out by the accrediting bodies. And we also need to version all patient reports so that all software and methods that we're using are correctly attributed to the version that they were calculated under, so we can go back and redo the calculation, in the future days, knowing what software version we used. And of course, we need persistent data for 2 years, even through database updates. We also need to have quality metrics available for reagents, instruments, data, process, and analysis. And we have to have our workflows, which I'll talk about in a bit, but our workflows have to be completely customized to our indication and platform, because workflows for PCR are not exactly the same workflows for next generation sequencing. So your laboratory information management system should be flexible enough that your workflows can be tailored and adapted. So let me go back a bit. So developing a custom LIMS, when we considered, actually, I think I jumped forward too far. Let me go through this one more time here. Let's see if I can get to, there we go, commercial LIMS for clinical PGD. So consider, if you're thinking about having a LIMS system for PGD in your laboratory or going to a commercial system, or even if you use a commercial system already, some of the things that you have to think about is how the system has been validated in different use cases. And if it's a good company, they'll give you lots of examples of how it's being used in a good clinical laboratory and how they keep up on validation, that the data is, for data integrity and for use. They can give you lots of help and information on how that's being done. if it's a system that hasn't been used for PGD but for other laboratory environments and you like the system, you have to ask yourself, how configurable is this system for PGD? What ongoing support does the software require? Sometimes it's not as simple as just unrolling the software and installing it. There's sometimes an ongoing support needed for both, correcting mistakes or extending the software to do what you need to do. So you have to ask, what ongoing support does the software require and what kind of costs are required, and how much work has to be done to completely customize the LIMS system to actually work for PGD and for all laboratory uses in a PGD laboratory. And cost for upgrading or additional customization and growth, that's also a concern. And also, how easily does the software customize for non-standard genetic tests in PGD? And ultimately, does the LIMS database allow transparency? So even though the LIMS software is sitting on top of a database, are you able to access that database cleanly from using external tools to actually do due diligence on the database or other types of analysis on the database that you don't need to go through the LIMS system to do? So doing third-party checks or doing database integrity checks, do you need to go through the LIMS to do that? Maybe sometimes the LIMS has things that built in that you're able to do that independently. I mean, it's built into the LIMS system itself. So this is the kind of questions you should consider. For us, we decided to develop a custom LIMS. And part of my job is to enable research here at RMA, New Jersey, by creating systems that make it very easy for research to go smoothly as possible. And I found that a lot of the commercial software we looked at would also require a bit of programming, basically, or wrapper writing. There's no out-of-the-box perfect LIMS system that can come out for PGD, especially for a group like ours where we do a lot of data validation, method validation, and then put it into production as a clinical laboratory. So we developed a custom non-commercial LIMS that we paired with our validated bioinformatics pipelines for microarray PCR and for next-gen sequencing. So for the front end, we built it in CGI Python. We drive our web interfaces in R. We use the programming R and Python to actually drive the analytics. On the back end, we have an automatic set of pipelines that do quality control and analysis. So the model is that the pipelines automatically do all the data acquisition from the machines, upload everything to the databases, calculate the results from the database, keep a record of all the lot numbers, all the required metadata, like who did the analysis and when. All that data is stored in the database, and at the front end of it, when the calculations are complete, you can visualize and browse the data according to any metric that you want to look at. We have several different choices. You can also look at lab performance from the raw data. So we store the raw data, the derived data, and all the metadata, and it all talks to each other. So it allows us to have a lot of control over quality in our data sets, or at least looking at quality in our data sets. So we developed it in an iterative fashion, almost in an agile software fashion, where we did a kind of an onion skin. We had a series of requirements for functionality as a backlog, and we started adding them on to our LIMS system as we went over the period of a year. So we started with a core functionality. We had the back-end processing pipeline, the results presentation, and sample tracking was the most important stuff that we needed to put in first. And all the front end, where we actually did the sample tracking and clinical diagnostic systems. So the first part was more like an R&D part in the first stage. The second stage was the finished product. And in the third stage, we're adding in the next generation sequencing pipelines for copy number analysis of embryos and also single gene disorder analysis. So building it up in a sequential fashion made it a lot easier for us to develop on the fly features that were required or only discovered that were required later on. So in our PGD data structure, we have essentially 3 different arms of the PGD data structure. We have the patient information that comes from the clinical requests. We have our laboratory entity records that are part of the actual laboratory function, and then we have our analysis records, which are the results of the statistical analyses. So when we check in, so for RMA genetics, when a patient requisition is checked in, we have all the patient information, independent of the actual samples that are run. Those are usually sent to us ahead of time. So here's a patient, she's going to have a cycle. It's usually focused around the female. She's going to have a cycle. We keep her information, her date of birth, and other identifying materials, and when her cycle will be. When the samples do come in, we have the embryo records are accessioned. We check them into our laboratory. We also, at the same time, have our instruments, reagents, and lab metadata ready, so that when the embryos are checked into the lab and put on a plate or actual laboratory entity, such as a plate or a chip, those embryo records that were accessioned in as samples that came into the lab are now linked directly to the chip or the entity that's doing the analysis. So it would be like a PCR chip, an NGS sequencing chip, or a microarray analysis chip. So those entities have their own IDs and are tied directly back to the embryo records. And in that case, because we're managing the laboratory entity records as a separate database, those entity records also connect to our instruments, our reagents, and a lab metadata. And that entire laboratory entity environment then can also connect out to the analysis results, which are done. So the analysis results are accessioned or are able to be foreign keys, both between the embryo record that was checked in that's connected to the patient, the laboratory entity records, and also the analysis records. And you can actually access this, and I'm leaving out a lot here too, but you can access any one of these records from the other records. So for instance, you can say what were all the samples run on instrument X, and what were the performance on instrument X on a certain date and time. What reagents were used, what reagent lots were used, and how do the plates and the embryo samples compare between the reagent lots on average. You can look at lab metadata, what particular operator was doing, what person was doing the analysis that day, or by date, or by operator, and you can see how the different plates and chips performed under that one particular person. So we have a lot of control to see how the data is and how the lab's performing in our system. So when we designed the LIMS, we had different considerations. When you design the LIMS at all, you have to consider whether or not your data structure and workflow should really be designed together. It's hard to design your data system first and then build a workflow on top of that, and vice versa. It really should be hand in hand. We did data testing and validation designed into the system. So the system has as much testing and data validation built into it as the same amount as the code that's built in to manage the data. So a lot of our data is done on a testing base. A lot of our development was done on a test base, which I'll talk about in a bit, on a testing basis. We maximize our automated processes on the database so that the workflows are completely automated. And we actually over-design for all possible metadata needed to reproduce results. We try to version control whatever possible, and we plan for growth. So there's a lot of modularity in our data, as you saw in the slide with the ovals. A lot of those databases are modular, and a lot of the workflows around them are modular, so that we can change any one of those without having to change the entire system. And a lot of good LIMS are probably built like that in the commercial world as well. We also over-provision the database and system hardware. So we try to buy more than we need because we're always growing, and we don't want to have to suddenly say, oh, we need a new system because it's getting too tight. We want to have a lot of advanced notice for that. So we over-provision it. Now here's a slide that was skipped, and I know it's kind of hard to see, but this is an example workflow for the data that we do for a typical PGD job. So this is checking the patient data, moving to different job IDs, sample IDs, how the sample's handled. We do video recording of different when samples are transferred. We do video recording of the sample to make sure it's adequately done. It's reviewed by a laboratory supervisor. We go through and the data is analyzed, and there's electronic signatures from a board-certified cytogeneticist. This is all automated, and there's very little human interaction except for the beginning of the pipeline and the end of the pipeline. There's a couple of different check-in states, but usually they're automated, and it's a binary, is it checked in or not kind of setting. So we tried to take out the human element as much as possible without completely removing it from controls. So there are some steps where a person could step in and change something, but on standard operating procedure, that's not done. So when we developed the LIMS, that workflow was done on a test-driven development. So we checked on quality, reproducibility, and correct exception handling. In that, when there's errors that were found, when there are errors that are found, we record them. And if there are problems with the software, it doesn't just crash, it goes and does something appropriate. So if for some reason an incorrect value slipped through or something came off the machine that was unexpected, which sometimes happens, the data should not simply just drop and the sample should not just disappear from the horizon. What should happen is there should be an automatic email with the LIMS system saying, excuse me, there's a problem with this data. It shouldn't just fail. So there's a lot of exception handling that was built in to our LIMS system. The tests were defined up front before the code was written. So we said, well, okay, we need to test to make sure that the sample, when it comes off the machine, is correctly entered to the database. There isn't a crash. So in that case, you would define a test up front, and when you go and test it on a sample, your LIMS system will always have that test in place. So you've built your code around that test. And so there's other tests that I have here. We define tests for almost every function in our laboratory information management system. And the code was written around the tests, so they're successfully completed. And the tests actually remain as part of the LIMS, so that we periodically run those tests to see if everything's okay in our test situation. And if there's any kinds of changes to the system or changes to our instruments, if there's failures of our tests, it indicates a documentable problem. And there's often, our machines are often updated by the manufacturer with new software versions. And more than once we've caught with our LIMS tests, for instance, the loss of an instrument ID being put out, or different challenges with the changes in error reporting on the sample output from the instrument. All those things actually could change the way our LIMS system was operating, and the tests that were run caught those. So quality checks and LIMS development, I talked about that. Okay, so automatic workflow tests. So one would be job process. Job accessioning, every month we have an automated system that creates a job ticket. It tests all exception handling, so all the things that could go wrong when data is entered. We run that through a simple test, and then at the end of the test, we should have a successful job registration with the right data in it. And then it comes back, and if you look at the monthly report, it'll just say job process entry okay, because the okay means everything is completed successfully. If it doesn't complete successfully, on the report it'll say not okay, or actually it'll say exception on it. For analysis test processes, we run raw data from 3 embryo samples of known status per month. You can run them through. You create data. The process will create database entries. It'll send raw data through the platform pipeline. And it's not fresh data off the machines, it's data of known characteristics. We kind of have benchmark data. And the final analysis result should replicate against that previous benchmark. So you should get the exact same result out no matter what changes have been done in the LIMS or in the data or in the reference, anything like that. You should get expected results out down to the decimal point, and that's one way of running a test. Another test that we can run is laboratory test analysis performance per month. So summarize all the samples run that month in a certain test, and you can flag any deviations beyond some certain threshold. So some of that would be population, of sample population, comparing a previous month that you set. Okay, so for 2012, we're going to always run February, for example. Check to see that February, when we do our summary analysis, February always comes out to the correct value. So you'd run February as a benchmark, and every month when you run your test, also run another test with expected results. So you're not going to show that, but you'll say that that the test parameters you ran should be expected because the one that you knew about how it should run came out to the same value. So that's another way of doing a test analysis performance. So for copy number visualizations, I want to go a little bit into visualizations. Unfortunately, I'm not going to have a lot of time to go completely into visualization like I wanted to in my abstract. But there's certain things in our LIMS system when it came out with reports and for quality control measures. Our copy number visualizations had to be accurate to produce rapid and accurate review of views and reviews of results. Chromosomal copy number analysis is essentially looking at chromosomal counts per embryo. So if there's a trisomy 21, you should have 3 copies of 21. If you have a monosomy 6, you should have 1 copy of 6, and it should be obvious in your visualization that that's what you have, so that when your cytogeneticist is looking through results, he or she not only has the table in front of them, but also a visualization that's accurately reflecting what's in the table. So it should also include pertinent information, like the patient information, directly on the chart, and that should be part of the LIMS. You shouldn't just have a chart on top, with, to one side with the data. The data should be put on the chart to show that when the data analysis was run, it was run on that particular patient, that particular sample. So that should come forward with the chart. It should convey complex information in easy-to-read layout. It should include validated measures of variation. It should include decision thresholds on the chart. Also, it should provide alerts on the chart where data is outside an acceptable range. We use asterisks and bolding, but you could use whatever you want, as long as it lets the reviewer know that this data is somehow outside of the nominal range. We also provide multiple detailed view types for the same data, and that often helps because sometimes one view of data isn't always the best view of the data. So here's an example of one copy number testing visualization we use, and here's a cell line sample of +16 +21. This chart is just an R, and the bars indicate the chromosomes, and you can see that 16 and 21 are elevated above the blue bars, which are empirically derived cutoffs for those chromosomes, and they're copy number 3. And you can see X and Y there are monosomies, meaning this is a male. It's a sexed male sample, and those were our monosomies. And the little numbers above each one is the z-score deviation from the normal reference distribution of copy number 2 chromosomes. So it helps in decision making for the cytogeneticist. So this particular view is supported by another view we've done with heat maps. And here I have an example of that was recently published by our group, where we were able to use the heat maps to identify incidental balanced translocation carrier patients. So the heat maps are normally used as another view of our assays and how at least in PCR, the PCR that we're using, our assay responses to copy number. And in this case, if you have a trisomy, it tends to be very bright white. And if it's a monosomy, it tends to go down to red. So, and think of hot iron, going from deep glowing red to very bright glowing white. So that heat map, so most of the orange colors mean copy number 2. It's in the median range. In this example, we have chromosome 10 and 13 have a translocation where part of 13 is, in these cases, unbalanced translocations, so parts of 13 are lost and parts of 10 are gained. So these unbalanced translocations also, you would have not been able to catch that in the previous plot. It would have just shown that some of the data's variation was out of range. But by using this plot on the plate surface, you can actually see the locations of the translocations, and that was something that we actually have seen more than once. But this is one example that we recently published. Okay, so that's the visualization part. I have more visualizations. Unfortunately, I can't show them yet because some of the methods we're working on is still not published. And if you look me up next year, I'll have some really interesting visualizations for copy number and next-gen sequencing. But I can't show you that right now because of the intellectual property issue and the pre-publication issue. So unfortunately, I can't do much more than that. For pre-implantation genetic diagnosis by next-generation sequencing, I'm going to talk a little bit about this. I wish I could do more, but again, we're working on this still, and we're in the process of finishing up our protocol, so I can't talk about it yet, but you'll probably see this in print sometime soon. But I will talk about the process. Our sequencing pipeline, most sequencing pipelines go along this process where you have your data from your sequencer, you do sequence retrimming and filtering, sort of a pre-alignment conditioning of your data. You do an alignment to your reference. You do alignment refinement if you want that alignment to even be better aligned. There are software that will do that. You do some sort of variant calling if you're looking for particular gene variants, and you can do variant and statistical analysis. If you're doing copy number, the variant calling part doesn't necessarily have to happen. Other statistical analyses will happen at that point, which will yield copy number in your data. Now in CAP, we talked about earlier in the presentation how CAP has now got a new bioinformatics pipeline for next generation sequencing requirements. They also have a section in there for maternal plasma next generation sequencing for fetal genetic tests, where you can actually, shorter DNA reads that you actually obtain from maternal blood can be used to identify are assumed to be fetal and embryonic DNA that enters in the maternal blood. And interestingly enough, you can use that to do some genetic diagnosis on the embryo, and you can actually do copy number analysis as well and sex the embryo. So all this can be done in maternal plasma, and even though there's not a PGD specific set of requirements in CAP, those interested in maybe what could eventually come along as requirements for PGD, at least through CAP, you might be able to see some of that in the next gen sequencing for fetal genetic tests. For the bioinformatics pipelines for NGS requirements, this happened in July, and last July in 2012, and CAP has actually had a working group, I believe, together that got together and determined what kind of information would be necessary to support clinical use of next generation sequencing. So including in that is the documentation, revalidation of the data analysis process, quality management programs, any kind of update policies, what happens when the software is updated, what kind of storage policies do you have for the 3 main files that people often use for next generation sequencing, including FASTQ, which is your raw read data, your BAMs, which are your alignment, binary alignment files, and your VCF, which are your variant calling format files. So all those files are usually, they're quite large. BAMs are slightly smaller than FASTQs, and VCFs are slightly smaller than BAMs, but they're all very large. So we should have versioning traceable for each patient report. And we should have storage policies. Pardon me, I'm having problems with my phone. I'm hanging it up. We also need exception logging where pipeline processes vary from the standard operating procedures, and our data transfer confidentiality policy has to be in place. All these are requirements in CAP. I have the link there at the bottom. If you want to do NGS, I suggest you read this before you embark on it, because if you plan on doing accreditation through CAP, because it's actually pretty intelligent, and I read through it, and I think it's the right way to go, at least with the beginning of accreditation of next-gen sequencing. So for the last few minutes, I want to talk about applications of pre-implantation genetic diagnosis and next-gen sequencing in human genetics. But before I go much more into that, I want to talk about what's already been done. So there's been several markers that people have worked on for clinical labs and rapid discovery and next-gen sequencing. But there aren't any clinical trials right now that have been validating the use of NGS for PGD on a laboratory basis for single gene or copy number analysis. Meaning that there's no clinical trial that's helping to set up a standard. So methods actually have been used, though, to identify deleterious changes in a genome. So there's been in one paper as an example where sensory autonomic neuropathy caused by mutation of dystonin, where I believe an exon of dystonin was missing in affected families, and was recently published in 2012. And part of that was done through loss of heterozygosity analysis coupled with whole exome sequencing. And of course, this had to be validated, and it was published. But things like this method of rapid discovery through next-gen sequencing, and then validation, which is an important part I don't mention on the slide, but I have to point out that there are a lot of variations that could be responsible. You have to validate, obviously the variants have to be validated as causative. But once that's done, the discoveries can allow for selection against effective carrier status. with PGD testing on embryos. And this can go, and there's a lot of different ways that this can be applied. Specifically for more complex disorders like autism or adult onset diseases, we may eventually get to the point where we'll have the opportunity to select embryos who are less likely to get certain diseases. So in the workflow design considerations for next-gen sequencing, if you're thinking about it, it has a unique set of requirements for analysis. If you remember the earlier slide I showed with the bunch of the workflow for NGS, each step could be a different piece of software that is often rapidly versioned. So NGS workflows have not quite reached commercial maturity since most of the software is publicly available. It's because they're academically designed, so the rate of development and change in the software is pretty high. Some of it's pretty established, but there's always room for improvement, and it's still a developing method. So especially as the data density gets bigger and bigger, there's going to be more and more software that comes out to manage the data. So that's a design consideration in your whatever platform you use to do NGS sequencing. Now, PGD applications really require tailored specific parameters to optimize the results of next-gen sequencing. It's not as simple as just throwing it against a reference and seeing what you get out. There has to be some statistical analysis that's not going to necessarily come directly out of academic commercial software. So you're going to need some statistical analysis support from a good statistician with experience, especially if you're doing methods development. And because there's also no one-size-fits-all analysis solutions. So some examples that I have here are that for mitochondrial heteroplasmy analysis, if you're interested in doing that, it hasn't been for research purposes. You have a small reference, about 16,000 base pairs. And to look for heteroplasmy, you have to somewhat relax parameters, and your data is a small to medium data size. So you don't have really large data, but your parameters in your alignment software are going to be different than if you did targeted gene disorders, where you want to have pretty strict alignment requirements. You don't want to align junk to your reference, but you also have a different requirement for chromosomal copy number analysis, and you'll have yet another requirement for variant discovery. So all those requirements are different settings in your software that if you use these as a laboratory test, you're going to have to record. They're all going to be different parameters, all recorded separately and differently for each method that you use. So it's almost like you have a different assay or a different platform, even though you're using the same platform at NGS. The software and the requirements in that entire pipeline can be completely different between two applications. So it's no one size fits all, and please keep that in mind if you're designing your workflows. So choices for NGS-PGD workflows. You know, your choices are, if you want to set up a workflow, is you can only do simple. You could stay with the simple analyses like single gene disorder type of analyses where you have a small reference and a targeted amplification. And often you can probably find a commercial vendor that'll support those results or support that pipeline. Or you could probably hire a bioinformatics or data analysis expert professional to research and design workflows that are a little bit more complex for you if you're doing research and development coupled with clinical testing. Or you could hire consultants. There are plenty of good teams out there that are bioinformatics teams that can help you put in place a pipeline. I've worked in organizations that have done that before, where the pipeline would be more of a work-a-day thing. They can come in, install a pipeline, validate it for you. It costs money, but they're available often for updates and upgrades. So if you have a good relationship with that kind of person or that kind of group, it can be useful for your organization, and you're not often tied down to one particular product. And they have a lot of experience, so you leverage that too. Or you can wait for commercial software out-of-the-box developers to catch up with NGS development and hope they include your method of interest, whatever you're interested in. But ultimately, in all cases, the rate of development for NGS is very high. So any established solution you put into place may change both in the platform that it's being used, or there might be upgrades in data density, bigger chips, more accurate results, error rates that are different from platform to platform. So anything that you put in now may be different a year from now. So that's another thing you should consider. So if you do consider building this or partnering with someone or using commercial software, try to figure out if it's going to be able to be used a year from now, or if you project that it could be used a year from now. And a little bit in the last 5 minutes I have here, I'm going to talk a little about human genetics and future pre-implantation genetic genomics directions. Really, we're all about assessing reproductive potential here at RMA, and we're interested in thinking about where PGD might go, even if our considerations are way out there, like where might it go in the future. So to understand where we are in terms of human genetics, there's been two main efforts to catalog genetic similarities and differences in human beings. Because in order to understand where genetic diagnosis is going to go, we should understand the background of what we're operating in. So in understand the typical genomic landscape and genetic landscape of the average individual, to quantify typical variations, there's the HapMap project, which began back in 2002, and it surveyed about 1 million SNPs per individual. These are single nucleotide polymorphisms that help both say something about genetic diversity and also about inherited conditions that individuals may have. Most of these HapMap individuals did not have any inherited conditions associated with them, so it's more of a population analysis level that we're looking at here. So they took hundreds of individuals from about 8 populations at known variant locations and tried to see what kind of genetic background were in each individual population, which I'll talk about in a bit. There's also recently the 1,000 Genomes project, where it published an integrated map of genetic variation for about 1,000 different human genomes. And they sequenced billions of genomic locations per subject over 14 populations. And that is available now publicly if you're interested in looking at it. They recapitulate some of the HapMap project individuals and additional individuals, so you can compare the two if you're interested. They did discover, or it seems to be dropping off here, but what they discovered was about 38 million different single nucleotide polymorphism loci in the human genome, about 1.4 million insertions or deletions that were less than 50 bases wide, and about 14,000 large deletions in functional genomes. So there's a lot of diversity just in the small amount of population that they did. Roughly about 1,000 people, which is it's definitely not the entire diversity in the human genome. It's just a fraction of it. But even in this fraction, we found an awful lot of diversity. So to give you an example of what I mean by diversity, here's a slide with an image from a paper up at the top if you want to go look it up. It's a great read. It's the 1,000 Genomes Project publication that was published in Nature back in 2012. And this is just 1,000 kb range across 2 genes. And in light blue is agreement with the reference sequence, so the human genome reference sequence, like HG19. So in light blue are the agreement locations in the background there, and white were single nucleotide polymorphism locations that were known. So these people have variations and that locations are known. On the right-hand side, going across the screen, is each individual. Each individual is very small. It's only a very thin line. I think some of the little black lines you see there are probably representative of a single individual. We have 4 populations represented here. We have the American Indian or American group at the top, then the African group below them, Eastern Asian, so that would be Japanese, Chinese, and Beijing, and I think Chinese in America. And then European, TSI would be Toscans or Tuscany, CEU or groups in Utah, Iberian, I think Finnish, and Great Britain. In African, we have Yoruban and African Americans in the Southwest, and I forget the LWK. And then we have Mexicans and other groups from America. So the genetic diversity, you can see, first of all, in African Americans, you have an awful lot of genetic diversity. And the reason you can see that is the little blue crosses, which look like little tiny blue specks on there, are rare variants with less than 5%. 5% in the population. In the bottom of the entire graph, you see little black tick marks. Those are novel variants that have not been seen before. In little green arrows below there, you can see where there's indels that are found in each individual. And the little blue tick marks are the rare variants that say something about some of the rarer variations that happen in individuals. So if you're trying to look for disease status or doing genome-wide association studies, these representative little blue marks are pretty much the kind of, if any of these are representative of a disease, you're going to need to sample a lot of individuals to get the power to see their effect. But if you want more details on this and others, you should go read the paper, because it's a really good insight into what kind of diversity we're up against in doing genetic analysis in the human genome. So the functional consequences of variation, apparently about 1.5% of the total genome codes for the genome's 20,000 proteins. And then we know for about 75% of those proteins, we know some information on their biological function, whether it's in a specific tissue or overall how they function. And that's actually kind of generous. It's 75% is you know, even if we know that it's expressed in a certain tissue under certain conditions, and we kind of know a little bit about that biological function, we can say it's a kinase, then we know something about it. But there's still a lot of genes we know very little about how they function in every tissue. Dr. Marr presented yesterday about some of that. If you want to go look up her presentation about the differences in expression of some of these genes and how functionally they may differ between different tissues. So there's a lot of variation, again, that we don't understand. So we don't know very much about every possible function of the proteins and interactions. And some variations in protein coding regions are deleterious, which we find that there should be a loss or decrease of the protein's function, or that's predicted. And if we disrupt the sequence that helps regulate when and where these gene products are actively produced, some of these variations may actually disrupt where and when those are produced by disrupting the translational machinery. And also, there's predicted deleterious variations that may have some significant, some or no observable consequence to health. So that's about the 1.5% of the genome that we see where there's exons and we may have variations that might be cause disease. But the other 98% of the genome, we don't really have an idea of how what the functional consequence of variation across the rest of the genome that is not exons that produce proteins. We don't really know how that affects the function of the genome. To tackle some of this, the ENCODE project, which was published back in 2012, was actually looking at functional DNA elements and systematically mapped regions associated with functional consequences of gene production and control. So they actually were able to assign some sort of biochemical function or role to about 80% of the human genome, or at least the 80% they looked at. But it doesn't necessarily include any kind of physiological function, like how it would affect a patient, or implication of variations or functions to the overall health of the individual. Only kind of a functional expected function on a molecular paradigm. So in ENCODE, they used one particular genome, and they were able to show that that a lot of these variants actually may cause a functional effect, and a lot of these are outside of protein coding genes. So when we're doing analysis on exomes to look for disease in our individuals, there may be things outside the exomes that are causing a particular disease or a phenotype that we're interested in trying to test for. So in that case, whole genome sequencing may become important. Targeted sequencing of exons may be limiting us in some cases to functional variants that are only inside exons and not outside. I'm going to skip this slide, but you can catch up with this at the paper earlier cited. But essentially, the take home on this is that humans may each of us may carry a few hundred potentially deleterious mutations that we've either de novo or inherited from our parents. And the implications of these rare variant findings are that, that the rapid growth of human populations have allowed many of these rare low frequency variants to spread between countries and between regions. And so because humans carry greater than an expected number of rare variations in the population, these rare variants are actually going to be isolated to small groups or individuals. So the power to detect these in particular disease states, if you're trying to use populations of people with certain diseases and do GWAS studies, as we've already learned in the past several years, it's difficult to do because you may not have enough power. So why attempt NGS-PGD? Well, one reason to attempt it is that there's the sheer number of human variant loci that I just briefly discussed, coupled with the elusive genetic origins of several disorders, suggest that there may be variations causing these disorders and subtypes that thought to be single origin. So we may want to do whole genome sequencing and do some R&D coupled with our clinical testing, because autism, for example, there's evidence that it may be a spectrum of disorders caused by different variations, because we haven't been able to nail down any one particular variation that causes autism. They could also couple single gene disorder screening with chromosome copy number analysis based on whole genome sequencing of embryo genomes. Sorry, I'm reading from the slide, but that's essentially that's how we could use it. And our PGD labs in the future that are not engaged in genetics research, because of the challenge with whole genome sequencing and trying to identify causal variants, I personally don't think that PGD labs are going to be engaged in genetics research on the level of whole genome sequencing unless they're doing genetics research. But if they're not, I don't think that they're going to be having a great call for whole genome sequencing in the near future, just because of the sheer statistical analysis and power that will be needed to identify causal variants, and of course the validation steps. So next generation sequencing going forward for most laboratories is probably going to be targeted. That's my take on it. And of course, target and specific whole genome sequencing analysis for copy number. So I just want to go through the people I want to thank. Mostly, we're a team effort here at RMA. We have we have research and nursing support to andrology support here. There are whole teams of people involved in making our clinical program successful, including our doctors who dedicate a lot of their free time to our clinical trials. We have clinical embryology, and we have some great embryologists at work. And of course, the molecular biology lab here under Dr. Nathan Treff, who runs the molecular biology side. We have a great team of people doing our analyses. And I have working for me Dr. Batzel Defcoda that helps me with the LIMS system and the research and analysis system. And that's pretty much my talk. You can, for continuing education credits based on the slides that were here, you can go to bioconferencelive.cme/CME-CE, and you can also go over my slides again in the future and take your CME credits that way. So I'm going to look at the Q&A and see if there's any questions. I don't see any. So if anyone has any questions, we have a couple minutes. We start a little late. So does anyone have any questions? Doesn't look like we do. Well, thanks for sticking through a little bit of a dry conversation, but hopefully next year if I give this talk again, I'll have a lot more information to show on next-gen sequencing and how we're using it for PGD. And thank you very much.